This video describes the extraction procedure for available metals by ICP-OES and available nutrients by ion chromatography. First, 2.5 grams of our fresh sample is weighed into a labelled 50 ml centrifuge tube. Then, 25 ml of our extraction solution is added to our tube. For available metals by ICP-OES, a solution of 0.43 molar nitric acid is used. For available nutrients by ion chromatography, a solution of 1% potassium chloride is used. We carry out separate extractions in separate tubes for each analysis. Now our extraction solution has been added to the tube and it will be shaken at 100 RPM for 30 minutes on an orbital shaker. Our samples have now shaken for 30 minutes and the contents of the tubes have settled. Now they need to be filtered for analysis. To do this, we need a syringe, 0.2 micron syringe filter, and auto-sampler vials. We use a 1.5 milliliter Versa vial for ion chromatography analysis, and a 15 milliliter centrifuge tube for ICP-OES analysis. The filtration procedure is the same for both types of extraction, but more sample is required for ICP-OES analysis. First, we take a syringe, and we draw up liquid from the tube. Be careful not to draw up any sediment from the bottom of the tube. This will block the filter. Next, we add a syringe filter to the end of the syringe. These screw into place. And we transfer some of our extraction into the Versa vial. There's a small shoulder on the vial. Filling to around this level is plenty. The vial gets capped and put into an auto-sampler rack, and the sample name and location are recorded on an Excel spreadsheet for the instrument software. Our potassium chloride extractions have now been filtered and added to the auto-sampler rack. They are now ready to be analysed by ion chromatography. The same procedure will be carried out for the 0.43 molar nitric acid extractions for available metals, but we would filter into the 15 milliliter centrifuge tube, adding 10 milliliters of filtered sample. This is the Thermo Scientific ICS 5000 Plus ion chromatograph. The instrument contains two parallel systems for analysis of anions and cations. The two bottles on top of the instrument contain the mobile phase, which is used to carry the sample around the instrument. Our filtered samples have now been loaded into the instrument auto-sampler, ready for analysis. Here we can see the instrument auto-sampler taking a sample. The auto-sampler brings your sample to this. This is called the injection loop. It has a fixed volume of 10 microliters. At the right time, the injection valve introduces your filtered sample in the injection loop into the eluent stream. The eluent stream is the mobile phase which is constantly being pumped around the instrument. After the injection loop, we have a separation column. This is filled with an ion exchange compound. The mobile phase and your sample are passing through the column, but different ions take different amounts of time to pass through this. This is mainly due to size and charge of the ions. After this, we have an electrochemical suppressor. This is used to lower the background conductivity of our sample. And finally, we have an electrical conductivity detector. This is used to determine the concentration of our sample. The higher the concentration of a particular ion, the higher the electrical conductivity it will be. This mixed standard is used to calibrate the anion instrument. It contains a known amount of our analytes of interest, and a dilution series is analysed in order to complete the calibration. Here we can see the chromatogram for the analysis of a calibration solution highlighted by the red box. This is a plot of time in minutes on the x-axis and electrical conductivity in microsiemens on the y-axis. We can see the individual peaks for each analyte as they are detected by the instrument. In the yellow box below, we can see the concentration in milligrams per litre for each analyte. The instrument software has been told the concentration of our calibration solution and the dilution factor that has been applied to it in order to calculate the concentrations. This is an overlay of the chromatograms of the 5 solutions that are prepared to calibrate the instrument. In the green box on the right hand side, we can see the calibration graph. The small crosses and orange square on the line of best fit represent the concentration of each solution. In this case, we are looking at the calibration graph for fluoride. Each analyte of interest has its own calibration graph. This is the thermoscientific ICAP 6300 ICP-OES spectrometer. It is used to determine metal concentrations in liquid samples. ICP-OES stands for Inductively Coupled Plasma Optical Emission Spectroscopy. Here we can see the inductively coupled plasma. This is an argon plasma. It is a high energy environment. At around 8,000 degrees Celsius, it is hotter than the surface of the sun. The optical emission spectroscopy is carried out by a charge coupling device. This is similar to a very high-end digital camera sensor. When metals in our solution are introduced to the high-energy plasma, they undergo a process called excitation, in which an electron is elevated to a higher energy level than its base state. When the electron returns to its base energy level, it emits light. The detection of this emitted light is optical emission spectroscopy. Each element emits signature wavelengths of light, allowing us to determine concentrations of individual elements. This is the instrument controller. The software on the PC is used to set the instrumental parameters for analysis. This is the auto sampler. It contains 2 types of sample: our unknown samples, our extractions, and calibration solutions, which are used to accurately determine the concentrations of our unknown samples. Our samples are pumped by the auto sampler with the peristaltic pump through to the nebulizer and into the spray chamber. The finest mist in the sample is introduced to the plasma, where optical emission takes place. These multi-element standard solutions are used to calibrate the instrument. A dilution series of these solutions is prepared to make a range of calibration solutions. The instrument software has been programmed so that it knows the concentrations of each of these calibration solutions, allowing it to prepare calibration graphs for each element and accurately determine the concentrations of our unknown samples. Here we can see the calibration graph for copper. On the x-axis we have concentration of copper in parts per million. On the y-axis we have intensity of light in counts per second. The instrument software calculates the concentration of copper by assigning values from the intensity of emitted light in our calibration solutions, which contain known concentrations of copper, and plotting a calibration graph. The red crosses on the line of best fit each represent calibration standards, which contain 0, 0.5, 1, 2.5, 10 and 25 parts per million of copper. The instrument software then uses the straight line equation y equals mx plus c to determine the concentrations of our unknown samples. In the red box on the right hand side, we can see the wavelength of light at which copper is detected. This is the same wavelength of light that copper emits when it undergoes excitation. The sensitivity of the instrument can be noted here. The sensor can differentiate between wavelengths down to picometer level. In the green box below, the calculated limit of detection for copper is displayed. It is 0.002 parts per million, which is equivalent to 2 parts per billion, or 2 micrograms of copper per kilogram of sample. In the red box, we can see the calibration graphs for our other analytes. There are a total of 18 separate calibration graphs for each element present in our calibration solutions. In the green box, we can see the expected and detected concentrations present in our 6 calibration solutions. In the yellow box, we can see the element that each graph represents and the wavelength of emitted light that this correlates with in nanometers.